Hello viewers, in this video we are going to know about flower development. First of all, let us know what is the terminology related to flower. So inflorescence meristem is an important term here. You should know what meristem is. The growing shoot tip during the reproductive flower producing phase of development is known as inflorescence meristem. What is it? It's actually a re-modified or repurposed shoot tip which will ultimately develop in inflorescence. So the shoot tip or meristem in the shoot tip during the reproductive and flower producing phase is known as inflorescence meristem. And what is a whorl? A concentric ring of floral organs, right? Outermost, we know that there are sepals, then inner whorls are there like petals, stamens, and then ovaries there, carpels are there, right? So, concentric ring of floral organs is known as whorl. Now, what is perianth? The outermost sterile floral organs that include sepals, petals, and tepals. I'm sure that you are aware of what are sepals and what are petals. And what are tepals? When the organ of the flower perianth, which is actually the first and second world organs, which are same in form, then they are called as tepals. So what are the tepals? First and second world, that is the sterile floral organs, which are in the same form, will be known as tepals. For example, if sepals and petals both are green color exactly looking, then these world will be known as tepals. Then ovules, the organ within the carpel. What is the ovule? The organ within the carpel that contains embryo sac and develops into seed is known as ovule. Then floral meristem. What is floral meristem? A group of cells that is initiated by the inflorescence meristem to generate the organs of flower can be known as or can be called as floral meristem. The vegetative and reproductive structure of the shoot are all derived from the shoot meristem, right? During which is formed during the embryogenesis. And there is an evolutionary conservation of flower genes and common patterns of flowering regulation. There are conserved genes and patterns for that regulate the flower development. And a signal from the leaves move to the shoot apex and influence flowering. Actually, signal from root, signals from shoot, and signals for even cotyledones travels to the shoot apex and at the site of towards the side of flower development and affects deflowering. In some species, the flowering signal is a response to environmental conditions. We know that environmental conditions like temperature, photoperiod all affects the flowering in plants. The developmental pathway leading to flowering are regulated at numerous control points in different plant organs, which are root, cotyledones, leaves, and shoot apices. In various species, this vary, right? In some species, major signal will be coming from leaves or shoot apices or from root, right? And also they vary in the flowering time and reproductive structures or architecture. Now, in case of woody perennials, there is a phase known as juvenile phase, during which the plant cannot produce reproductive structure, even if all the appropriate environmental signals are present. Okay, so if environment is fine, photoperiod is okay, temperature is okay, everything is fine. But unless that juvenile phase is not crossed, the 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 plant or the tree won't be flowering, right? So there is a particular boundary in terms of time when a plant will be or tree will be in vegetative phase and then followed by a reproductive phase. Vegetative to reproductive transition requires signal and a proper balance of inducive and inhibitory signals is required. A proper critical balance is required. So meristem, which is competent to keep making vegetative structures or may be induced to flower, make do the transition from vegetative to reproductive phase. So this meristem competency determines whether or not plant will be responding to signal. So it should be competent enough to respond to that signal. Either it is floral promotory signal or floral inhibitory signals. Leaves may also need to develop competence to respond to environmental signals before they can produce floral promoters or inhibitors. So the leaves are also required to be competent to receive the signal from environment and then uh, to promote the floral development. The structure of phytochromes also is modified by light, red and far red light, and these changes can initiate a cascade of events leading to production of either floral promoters in green arrow or the floral inhibitors. Right, leaves, cotyledones, and roots have been identified as the sources of floral inhibitors in some species. As I told you that a critical balance between inhibitory and promotory signals is required for reproductive transition. Vernalization. Vernalization is a period of chilling that can, induce flowers or make the flower shoots to competent to perceive and promote flowering, right? And you must have heard of many applications of vernalization. Through vernalization, we can change biennial plants in we can induce biennial plants to flower early and for fruit set, right? So it will reduce the time that we spend for harvesting fruits. And also flowering is also induced by grafting the vernalized shoot apex with that of non-vernalized one. So if you have a shoot apex that you have vernalized and you can transplant it onto a non-vernalized one and you can induce flowering in a shorter period. And also this feature is used in horticulture and the crop plants of winter varieties which can be subjected to spring vernalization and can be converted into a spring variety. So winter varieties can be converted into a spring variety by using this vernalization process or chilling process, right? So here you are seeing a grafting experiment where Nicotiana tabacum and Nicotiana sylvestris, which are two different plants in terms of the photo period they require to flower. Nicotiana tabacum is a day neutral plant that flowers when meristem gains competence to respond to the internal signals, while Nicotiana sylvestris is a long day plant that flower when the floral signals reach a critical level, right? So Nicotiana sylvestris, that is NS, is a long day plant. Nicotiana tabacum is a short day plant. If you see this figure here, there is a scion, which is shoot that is grafted onto the stalk, which is the rooted portion of the plant. Okay, so scion and this stalk. So here the stalk is flowering Nicotiana tabacum, means it is already competent to produce flower, right? And this one, the stalk is the flowering Nicotiana tabacum. Again, the stalks are same here, but the scion that has been used here is a young Nicotiana tabacum in case of first figure, and it is an older one. So if you see before flowering, it grows up 21 new nodes. So longer time is required for this young Nicotiana tabacum cyan to flower. Although this was placed on the flowering Nicotiana tabacum, and this older cyan developed only 11 nodes before flowering. Right, so it was competent to receive the signal from this flowering Nicotiana tabacum stalk. Likewise, if you see here, these are the stalks of Nicotiana sylvestris. One is the flowering one, and another one is the young one. So, flowering stalk is capable of sending signal to the scion of the Nicotiana tabacum quickly, and only 11 new nodes are formed before flowering. Right here, the young Nicotiana sylvestris takes time until the floral signals reaches a critical value and then sends a signal to the Nicotiana tabacum scion to grow a flower. Right. Meanwhile, it grows up to 25 new nodes. So this way, getting competent and critical level of floral signals is required in a plant to flower. All the life processes are controlled by genes, right? And flowering is not an exception to that. Many genes control the flowering in plants, and we need to understand that after a period of vegetative growth during which the plant maximizes light capture for photosynthesis, a combination of endogenous and environmental cues induce flower development. During this flower transition, the shoot apical meristem becomes a inflorescence meristem, which produces a distinctive architecture of secondary inflorescence meristem followed by the meristem for each individual flower, right? So a shoot apical meristem becomes inflorescent meristem, then further division or further differentiation into secondary inflorescent meristem, and then further specification or specified individual flower region are specified on the meristem. The flower development occurs in sequential stages. In first stage, floral meristem fate is specified through the activity of the floral meristem identifying gene or floral meristem identity genes. So again, the genes are the one that floral meristem identity will be decided by. Okay, and second is the floral meristem is patterned into the world of organ primordia through the activity of floral organ identity genes. Okay, first of all, floral meristem identity gene and then floral organ identity genes. Third is the floral organ identity genes activate downstream effectors that specify the various tissues and cell types. So now once the floral identity genes are active, floral organ identity genes are active, genes will be activated for specifying tissues and cell types that constitute different floral organ types. Induction of floral meristem fate is actually controlled by multiple pathways that respond to different environmental and developmental cues. Floral pathway integrators that activate floral meristem identity genes such as leafy, LFY, and epitella in Arabidopsis thaliana and their respective homologues, floricula and squamosa in Arabidopsis majus. Other factors that promote a floral meristem fate include cauliflower that functions, the function of which is completely overlaps that of Epitella 1 and Arabidopsis thaliana usual floral organs UFO, or as this Arabidopsis majus fimbriata. To a large degree, AP1 functions downstream to leafy. Functioning in opposition to leafy and AP1, Arabidopsis thaliana terminal flower 1, TEL1, or this Arabidopsis majus centroradials and AGL24 specify an inflorescence meristem fate. Okay, so there are different genes and regulatory elements that responds to different environmental and developmental signals and help identifying floral meristem identity or the floral organ identity. In this regard, a model was developed known as ABC model. So ABC model of floral world specification is actually dictates that there are certain set of genes that determine the or that give the region specific identity to different floral organs. So floral meristem identity genes initiate a cascade of gene expression that turns on region specifying or cadastral genes. So region specifying or cadastral genes which further specify pattern by initiating transcription of floral organ identity genes. So there are genes that determine the floral organ identity. This particularly Superman or SUP is an example of cadastral gene in Arabidopsis that plays a role in specifying boundaries of organ identity gene expression. Okay, so the boundaries will be defined by the Superman gene, which is a cadastral gene, or specify the region in the flower. Okay, So the flower organ identity genes were originally identified in Arabidopsis thaliana and Arabidopsis majus on the basis of their mutant phenotype. So several mutants were studied, and then later which gene is mutated and what it is causing. Like that, different studies resulted in identification of the genes conferring a particular phenotype in Arabidopsis species. And these genes featured the homeotic transformation of one floral organ into another type. So, particular gene mutation resulted in transformation of a particular organ, floral organ, into another. That's how we identify them. So, this ABC model postulates that three regulatory gene functions A, B, and C work in combinatorial fashion to confer organ identity in each world, like how the sepals identity is determined, or how petals identity is determined, or carpels identity or stamens identity. So a particular set of genes or particular genes are functioning in combinations to confer the identity of an organ. This model was formulated by George Hongan and Chris Somerville in 1988. Three classes A, B, and C of organ identity genes, according to this model, are really necessary to specify the four worlds of flower organs. You know that there are, in general, four flower worlds: sepal, petal, stamen, world of stamens, and then carpel, world of carpels, right? So, these three genes, according to this model, are required to specify these four worlds. These genes are homeotic genes, but not Hox genes. Okay, homeotic genes specify organ or specify regions in an organ or in an organism, while Hox genes are a subgroup of homeotic genes that specify identity of the organs in bilateral symmetry, or in organism having bilateral symmetry, right? So these homeotic genes in case of plants include AP, apetala, agamas, AP3, AP2, agamas, AP3, and pistillata in case of Arabidopsis. So we will shortly see that how class A genes alone specify sepal development, class A and B genes together specify the petals, class B and C genes together are necessary for stamen formation, and class C alone can specify carpel formation. You see here, this model is explained here. There are three genes, right? A, B, and C. So there is a region where only A is there. There is a region where A and B both are there. There is a region where B and C are there. If you see carefully, there is a region where C is only there. So A alone specify the identity of the sepal. A and B together, identify or specify the identity of petals. B and C together specify the stamen, and C alone is specifying the carpel, right? And one more special feature of this model is that A and C functions mutually antagonistically. So absence of A will be prevailed by the presence of C. Likewise, if C is mutated, then it will be taken over. The function of the C will be taken over by the A, right? So, and if A is mutated, then the function here, the region will be taken over by the products of the gene C, right? So A and C are mutually antagonistic. If A is not there, C will be there. If C is not there, A will be there, right? Like that. So A specify the identity of sepals. A and B together identify the petals. B and C together identify stamens, and C alone for carpel. And you see there is another class which is actually not the part of the ABC model, but there is a E floral homeotic genes which are expressed in all the which is actually a proteins minor protein, but it is expressed at all the floral organ levels. If A, B, and C all three are mutated, then only this E genes will be expressing, and all the floral organs identity will be changed to sepals. So So you see here, this is a normal looking healthy flower where all the whorls are intact and as per as they should be. And here, if where A is mutated, so if A is not there, C is there, and C alone is for responsible for carpel, right? So carpel, here are the carpillary tissues. So here you see, instead of having sepals, you have carpillary structures, and B and C together specify stamens. So stamens are present at their place, right? Again, B and C stamens, right? And then C alone is responsible for the ovary, right? So here we don't see any petals, right? Neither we see any sepals. So ovaries and stamens are the structures that we see in the flower, right? Here you see, if you carefully see, the B is missing, and both the first two words will be what? The first two words will be sepals, right? Sepals and sepals, because B is not there, and A alone will be making sepals, right? And C alone makes the carpels. So instead of having stamens, we have carpels here in case of stamens, and C alone again making the fourth word, innermost one, that is the carpel, right? So carpel again we have, and here where C is mutated, the so C is not there, then A is there. So we have alternating whorls of sepals, petals, sepals, sepal, petals, like that. There is no ovary, there is no stamen, because for stamens and carpels, C is critically important. Right, when all three are mutated, only sepals we see in a flower, right? This particular slide also shows you the similar thing. This is the development of normal flower with A sepals, with A and B petals, with B and C stamens, and with C alone carpels will develop. So here, if we have apetala gene mutated, so we don't have an A. So if A is not there, then C is there. So C alone develops in carpel, so carpel, and B and C make stamens, so stamens. Again, B and C stamens, C alone carpel, so this kind of flower. Likewise, we have different mutations, and then accordingly we see different kinds of flowers. So there are actually some more factors that regulate flower development, like ovule identity factors. A further class of floral D function genes. Okay, A, B, C, D functions genes, which confer ovule identity on tissues that develop within the carpel. Okay, so once the carpel is developed, what will determine the identity of an ovule? So these are D genes that confer ovule identity on the tissue that develops within the carpel. Okay. In Arabidopsis thaliana, ovule identity is conferred by a clade of four closely related genes known as AG, agamus, seed stick, and shutterproof 1 and 2. Okay. And AP1 and LIP1 and 2 are members of the plant specific family of transcription factor, that is AP2 and ERF, ethylene responsive element binding factors. See, not only environmental factors, but some hormones are also involved, especially gibberellins are known to affect flower development. Now, floral homeotic genes, including SCP, STK, and SHP genes, these encodes a MATS domain transcription factor. These MATS domain transcription factors have a specific region, and they form dimer and bind to specific region of the DNA. And these DNA binding domains are known as MATS domain binding regions of these transcription factors. I told you earlier that the sepatella SEP, or which we call the E genes, so these sepilata genes actually encode the proteins that forms tetramers that identify or that specify the identity of a particular floral organ. This quarter model is for potential formation of MATS domain protein regulatory complexes. So these are the protein regulatory complexes specified for that is specify sepal, petal, stamen, and carpel development. So for sepal, this quartet is of epitala 1, 2 epitala 1 proteins and 2 sepalata products. For petals, this is epitala petala 1 and 3, and sepillata 1 protein, and pistillata 1. Likewise, these quartet changes for stamens and carpal identity. Okay. These sepillata proteins functions as cofactors that provide floral specific activity to ABC genes by making complexes of their products, right? So these ABC genes products are complexed by these sepillata products for flower specific activity of these MATS domain protein regulatory complexes. Now let me tell you a bit about floral symmetry. In 1744, Carl Linnaeus, who described a naturally occurring mutant of toadflax, this is the toadflax plant, Linaria vulgaris, that altered bilaterally symmetrical flower to a radially symmetrical form naturally. This transition occurred naturally. So floral symmetry was changed from bilateral to radial symmetry. Linnaeus described this mutant as Piloria, which means monster in Greek. So we see different kinds of symmetry in flowers. We see asymmetric flowers, we see radially symmetric flower, we see bilateral symmetry, we see bisymmetric flowers as well. Asymmetrical flowers have no plane of symmetry, whereas bilateral flowers have only one plane of symmetry, while radial flowers have multiple planes of symmetry. If a flower has two planes of symmetry which are perpendicular to each other, then these are described as bisymmetrical ones. Symmetry types are established by the number of the petals and their corresponding angles. A flower with equal sized petals can create a radial symmetry when the petals are equally spaced, like these petals are equally spaced. Okay, so these form a kind of radial symmetry. Any deviation from these angles generate bilateral flowers. Sometimes, and as is the case in D and E, bilateral symmetry can also be created by unequal size petals in the flower, right? So these two are bigger and these two are smaller, but still there is a bilateral symmetry. The Antirrhinum majus flower shows a bilateral symmetry that is readily seen in the second word, which consists of two dorsal petals, two lateral petals, and smaller ventral petal. Okay, so we have a bilateral flower here of Antirrhinum majus. In this model species, the genetic program that establishes the dorsoventral flower identity from early stage of development includes the dorsal identity genes and protein products like cycloidea, dichotoma, and radialis. So in dorsal region, these are expressed. Okay, and this divericata in the ventral flower is involved in ventral flower development. And DIV is excluded from the dorsal domain of developing anterior major flower through the regulatory role of these red proteins or radialis proteins. Okay, so genes really play decisive role in development of the flower symmetry. I hope that you could understand this lecture or you could understand the flower development. I'm sure that you will keep coming and for new and more interesting videos. Thank you for watching this video. Do subscribe and like my channel.